Okay. Good afternoon, everyone. Welcome to the class on methods for studying imprinting. In the last class, we discussed some of the experiments and results. I'm unsure if we have already practiced a particular question that I usually have. The question is something like this. I will show you here. The CAA exam, in CAA exam, I could give you a theoretical problem like this. Okay. And did we discuss this in the class, in the previous classes? If yes or no, you please let me know. Can somebody tell? Did we discuss any problem like this?
No, sir, I guess not.
Okay, good.
But I
Then,
don't know what this is, sir. I'm not sure.
yeah, it is about imprinting and all. We haven't discussed, I think. So this is from a different class or last year's. Okay, it is about imprinting and southern blotting. If you remember what we have discussed in the last class, we were discussing about these things. We stopped somewhere here. Yes?
Yes, sir.
Okay. So we are continuing that in theory. So basically, what we are trying to read is about imprinting, and by means of DNA methylation as an epigenetic mechanism. And we were trying to use the restriction endonucleases because there are several restriction endonucleases which can consist or which recognize which whose recognition sequences have a lot of CG like this, or there could be others. So some of the restriction endonucleases are methylation dependent, methylation sensitive. That means they cannot cleave if the target site is methylated. Some of them can cleave and insensitive, and there are some methylation dependent. We are going to discuss about these two. Use methylation sensitive enzymes and methylation insensitive enzymes. Why is that? Because then we will be able to determine whether these cytosins are methylated or not. And this technique that we also have to discuss is based on southern blotting. And in that, we have to keep several things in mind, and that is especially of the probe, the location of the probe. And we should also know the genetic architecture or the genetic map. What is a map? We should know the distances of the genes or restriction sites that are present in the locus of our interest. So what we are going to do today is an exercise, and then followed by another technique that is used to study chromatin organization. So you can consider this as a tutorial where we are going to discuss several things, and this is likely to come for exam. So the first question or about imprinting that we need to discuss is about erasure and establishment. So from primordial germ cells, somewhere around the formation of primordial germ cells to the gametogenesis, right? Until gametogenesis. During the stage between somewhere around here, the imprints are erased. So majority of the times, those from that of primordial germ cells to until formation of complete gametes, there is no imprint, no imprints. But during gametogenesis, new imprints are made. That means if you look at a particular gene, and we also will get somatic cells at the end after fertilization, right? So if we have somatic cell or completely differentiated cells, and if we are talking about a gene or two genes, right? Because two genes and each, say, gene A and gene B, A and B, and each of them, one of which is inherited from the mother, the other from the father. So assume that gene A is expressed from maternal allele and gene B is repressed, whereas gene A is of maternal allele is repressed and B is expressed. That is a typical somatic cell. We will see completely differentiated ones or differentiated ones. But in the germplasm, there are some things happen. That is, say, the gametes, and between this stage, somewhere around here, we will see that there is, before the somatics, establishment, and then we will have here zygote. The zygote will give rise to eventually somatics, somatoplasm, and then the germplasm. The germplasm is what will give rise to the primordial germ cells. The somatoplasm here will form the whole body. The body, all the cells in the body will have imprints that have been established during the gametogenesis. Okay. Of course, here, if it is sperm, then in the other place it will be, um, one second. Yes, if it is, if gametes in one organism can form either sperm or egg, and if one has come from outside, and fusion, and then zygote. So from germplasm, this is the new germplasm. You can also think of life cycles. I think I have described you this way. Germplasm, and in the intermediate stage, we have the zygote, and the zygote will go through blastocyst and other organogenesis, and eventually give rise to another germplasm, and of course somatoplasm. Okay, and this germplasm again will give rise to another zygote, and this is the cycle that we know of. The important thing we also can remember, or is that, one second, I'll just try to take it in another way. Let me see if I can do this. So gametes, and gametes will give rise to eventually the zygote, and the zygote will give rise to primordial germ cells and a somatic plasm. All that the body that exists is the somatoplasm. That is what we physical body is. And primordial germ cells give rise to gametes. And this is the cycle. Simply two gametes, another gamete will fuse and keep on going. So from the time of the gametes formation, from the primordial germ cells are formed even when the embryo is in the womb. And by the time gametes are formed and released, it's about 13 to 15, or say 10 to 15 years. Maybe females will mature a long time, and all these years they remain dormant. And then gametes, and the cycle keeps on going. So somewhere during the prior to formation of gametes or so, there is no imprints. Imprints are erased. Erasure of imprints. And by the time they form the gametes, they become established. Imprints becomes established. Okay, that's the cycle. And what we are going to now learn is the techniques that are involved, and if we are trying to acquire the skill to interpret the data. We have already seen it. I hope this will serve as a good solution for when the question comes in the CIA. You will be able to explain better. I want to try Okay, let's go ahead with this. So I hope you remember this. Primordial germ cells, the standard model, at least the standard model. We have in the last class seen that not necessarily that all the genes of all the results from different labs have actually follow the standard model. One second, give me a minute. I want to see if, yes. So what we are going to learn now is if we want to see the model, if you want to see, if assume that you found out there are gene A and gene B which are likely to be imprinted, and you want to study how it is happening, then we would have taken primordial germ cells, or by going by the standard model, we would assume that around the time primordial germ cells to gametes time, there are no imprints. And then by the time they come to about the gametes, there are imprints are established. So we will try to build, keep those things in mind. And I would also want to make you recollect the southern blotting technique, right? So let's see how we'll go about it. So this is a genetic architecture, and each of them will have 2 copies, right? So I'm just building a new setup. One is from the paternal allele, the other one is from the maternal allele, and then we'll see if we can adjust this. Okay. Well, let it be that way. So assume that there is a gene. Okay, we will talk about only one gene for the time being. This is the gene X, and this is the same gene X. One of it is from the paternal allele, the other one is from the maternal allele. And I'm choosing a probe. A probe I'm choosing is supposedly it will bind to some region like this. Okay. And whether the gene is present or not, and the location of it is not really essential for us at this time. What is more important are the restriction sites that they would form. Assume that there is a restriction site there. Here is another, and here is another. So the same way, the other allele would also be cut like that. So you can think of it that the probe that we have here can also bind here because the sequences are same in southern blotting analysis. So if I cut the DNA using the restriction endonucleases, I would have to mention it. I will call it as this one as BGL2, okay? And this one is not one. And I also have to mention the sizes that are there between them. And the size would be, the size here is 3 kb, and this one is 2 kb. Okay. So the protocol in general would, or simplistically would be that we would have taken the genome, we would have isolated the genome, then we would have done the restriction digestion with BGL2 and not one, and then we would have allowed the electrophoresis, and I'll put it as southern blotting, during which time we would have used a probe, and I'll give a name, probe P. We'll call it as probe P. Okay, we would have hybridized it and developed the film. And what would we expect in general is the question now. So if you see the results of it, we'll have to draw a lot of lines like we did last time. And this question is very, I usually give it in the class, in the CIA exam for 5 to 10 marks. It'll be nice if you are in a situation where you are able to you are able to understand it and be able to interpret the data. Let me build this up. The best thing would be that if you have your paper with you, and I would actually suggest that if you have a paper and you are going to write along with me, then it will be the best possible way to for practice. Okay, the probe is somewhere binding there. I just moved it, I think. So I'll just put a few more lines so that it will be easy. Okay. So there's one more line we have to draw. And I hope you remember in some cases we would have drawn the line digested with only BGL2, and in some cases we would have digested with NOT1 and BGL2. So here are some samples. If I digested this DNA, I have taken somatic cells. Okay, I will just put it here as somatic cells. I have taken somatic cells, and I have to also mention something here. Assume that the gene X we are actually looking at is maternally imprinted. When I say maternally imprinted, it would mean that it was methylated in this case. So that is another piece of information you would need, maternally imprinted. Right? So here is, we have taken somatic cell DNA from the somatic cell, and we would have digested with BGL2, and in the another reaction where BGL2 plus NOT1. Okay, this is only with somatic cell. And I would like your participation in this and help me out fill up this blot. If I took somatic cell, how many bands will be observed? What will be after southern blotting? Okay, how many bands will we observe and what will be their sizes? Can anybody help me out? Yes. Maternally imprinted, when I said, it would mean that this allele here, say for example, is methylated. Okay, the green one that I'm showing is the methylation. So what band sizes would we expect? Anybody?
5
What will,
kb, sir.
yes, please. 5 kb, how would you get that? From where would you get that? Yes, you're right. But I just wanted the explanation.
Sir, in maternal gene, due to methylation, the not one site is repressed, and other two
Yeah,
are.
so not one site is uh, resistant.
Yes,
Okay,
sir.
yes, it is resistant to degradation by not one. Yes, you're right. It is protected from endonuclease activity. Yes. So we have a 3 kb fragment and 2 kb. Total is 5 kb, right? So the first one, the question is, I only said we will cleave only with BGL2. So what bands would we expect? And how many bands? Bands and their sizes. Anybody, please? I would like that everybody participated. The more you do, the better you'll get with these kinds of questions. Okay, I will try to help answer. And then, so BGL2 cuts here and here. And the same it goes with both the alleles. It has nothing to do with the methylation thing, right? So we will only get one band of 5 kb. Both cutting of both the alleles, maternal and paternal, is resulting only in a 5 kb fragment. Therefore, that is what we will see. If we look at if BGL2 and if you have digested this DNA with BGL2 and NOT1, what would we get? BGL2 will cut here, NOT1 will cut here, and BGL2 will cut here. So the fragments that should be coming would be 2 kb and 3 kb fragment, right? What about the maternal allele? BGL2 will cut here, and NOT1 cannot cut here because this region is immune or kind of protected by the methylation. And NOT1 is methylation sensitive. And BGL2 will cut here. So we will get, from the maternal allele, we'll get a 5 kb fragment. From the paternal allele, we'll get a 2 kb fragment and a 3 kb fragment. So that's what we can write. From the maternal allele, we are getting a 5 kb fragment, and from the paternal allele, we are getting a 3 kb fragment and a 2 kb fragment. You don't have to write these in your answers when you're answering in the exam. For the sake of understanding, I have written it here. Okay, this is so because we have taken from somatic cell. Now I'm changing the sample. The sample that I'm going to give is you're going to get the samples from primordial germ cells, PGC. So the one reaction is set where you are adding BGL2. I'll just write B, okay, for next time on. And in the other reaction, BGL4 plus NOT1. What bands will we expect? And what will be their sizes? How many and what will be their sizes? Anybody?
2 bands, 3 and
Yes.
2, sir, for
Why
both
is
alleles.
that? Why is that?
Because erasure of imprints take place.
That's right. During primordial germ cells, according to the standard model, we have erasure of imprints. That means whatever the green ones that I have written here as methylation is no longer methylated here. So BGL2, NOT1 will cut here, BGL2 will cut here. That same thing will also happen for the maternal allele. A cut here, a NOT1 will cut here, and the BGL2 will cut here. So both the fragments are going to be cut into, it will give rise to 3 kb and 2 kb fragments. And please also remember that I have given you that the probe is recognizing both the fragments, right? In the next example, we will
Sir,
make an alteration to this. Yes, please. Go
sir,
ahead.
for BGL2 alone, we won't get 3 and 2.
Oh, you are right. Yes, yes, yes. Okay. This is about just the BGL2, right? Yes, you're right. So I will just move these. Okay, sorry. Let's try again. Yeah, these two will are actually in this one, right? You're right. Thanks. So what will happen in BGL2? It will be only 5 kb fragment from both the cases. Okay. I want you to notice the difference between somatic cells and primordial germ cells in terms of when we added BGL2 plus NOT1. What is the difference here? BGL2 plus NOT1, you got a 5 kb fragment, 3 and 2. Whereas in primordial germ cells, you only get 3 and 5, 3 and 2. And there is no 5 kb fragment because during primordial germ cells, there is erasure of imprints and both the alleles would have been cut and 2 and 3 kb fragments would come up. So the next sample that we are giving is that we'll have is for, say, sperm. So this reaction is for BGL2 and the other one is for BGL2 plus N. What do you expect in this? Anybody?
Same as somatic cell.
No, just say what is it. I will then know.
We'll have 5 kb.
BGL2, okay, this is fine. Yes, you're right. What do you expect with BGL2 plus NOT1?
All
What?
three, 5, 3, and 2.
Why would that be? What do you expect in sperm to happen?
There's establishment already.
Yes. What should it be established as? Establishment of imprints.
Imprints.
If establishment happened, what would you expect? Would you expect that both the alleles are both the alleles are methylated, or both the alleles are unmethylated? What state would you expect? This I'm saying is maternally imprinted or maternally methylated. So what would you expect it to be in sperm?
Unmethylated, sir.
Yes, because sperm is from male, and in a male, the paternal allele is supposed to be unmethylated. So in that case, what bands would you expect and their sizes?
3 and 2, sir. 3 kb 2.
Yes, both of them, both the would be cut up, or anyway, in sperm it is haploid, and all this in all of these sperms, because there has been an imprint erasure and new establishment, and this gene is supposed to be in the paternal allele, it is supposed to be free of any methylation. Therefore, in all the cells, the not one will be able to cleave it. Okay, so we will get a 3 kb fragment and a 2 kb fragment. So on theoretical grounds, I want to have an alteration to this. If if this same thing or has given rise to, or assume that there is an egg we have got with similar from the same or different organism, uh, what would you expect in that case for the same setup? It's a theoretical question, so it's about we being able to understand the how to interpret and what to predict. It's about understanding the technique and the method and the interpretation. So what would you expect if it were egg? So we don't have to really speak about BGL2. There is not so much. Yes. Then
One 5
what would,
kb fragment.
why is that?
Because it is maternally imprinted, so
Yes. Say, come on.
it cannot uh, do endonuclease activities.
Yeah, the not one cannot cleave the DNA, right? So all in all the eggs, we would have, because this gene is maternally imprinted, in all the eggs, this part of it would be methylated, and therefore not one cannot cleave there. So we will get a bands like this. Yes, this is how we interpret data. So I still have, how many lanes do I need? 1, 2, 3, 4, 5, 6, 7, 8. I have 6. I want to have a change. I want to prove or work on this experiment again, and I want to see if what I said is right or wrong. But I'm going to, so I'm going to have a little bit of change, and that change is, I'm going to change, have a different thing. I'm going to have a new probe, and the probe is, sorry, probe is called, I'm going to have a new probe, and I'm calling that as probe R. The experimental setup is same as what we have discussed before. Okay, the only thing I've changed now is that of the probe R. Here, all the setup we used, in this case, we used probe P. Now I'm having a difference. I'm going to give it as probe R. What would you expect for somatic cells in B and B plus N, in primordial germ cells, B plus N, in say sperm, B plus, okay, B and B plus N. This should be B as well. So what bands would you expect in this case? Just there is only one difference, and the difference is about the probe. Okay, so what would you expect? Band sizes, and how many bands? If you say, then that will be fine. Anybody? For somatic cells, what would you expect?
5 kb, sir.
Okay, this one has no, yes, 5 kb. Then B plus N, what would you expect? Somatic cells. Anybody? So if in this case a cleavage would occur here, and whereas in the maternal allele there would not be any cleavage, right? Here there is a cleavage here from the paternal allele, but from the maternal allele, because there is methylated and we are talking about somatic cells, there would not be a cleavage. So we will get one 5 kb fragment that is from the paternal allele, and we will have one 3 kb fragment. We will not get 2 kb fragment. We will actually get 2 kb fragment. Only point is the 2 kb fragment that is here, okay, if it is this one, will not be detected because the probe is complementary only to this region. So when you develop the southern blotting, we will not be able to find out that there is 2 kb, this 2 kb fragment. So we will only be able to see the 3 kb fragment from the paternal allele. Okay. So whereas compared to that of P, the probe P that I have said, it is covering both the regions where the knot site is cutting. So in that case, it can recognize both paternal, both the fragments, 3 kb fragment and 2 kb fragment. So if you now with primordial germ cells, what would we expect? Is BGL2 and not 1? What would we expect? Anybody? In primordial germ cells, it would have erasure would have occurred. So we would
Only 5
get,
kb, sir.
yes, only
5 kb, sir.
why 5 kb? Can not one cleave or cannot? Can it not cleave?
It cannot cleave, sir.
Why is that? What happens during primordial germ cell stage or around that stage?
Erasure.
If erasure is happening, then methylation is occurring or methylation is removed.
Oh, sorry, sir. Sorry.
Yes.
Removed, sir.
Or yes. So what would you expect? That's all right. What band would you expect?
3, sir. Both 3 kb.
That's right. Because there is no imprint, because there is no methylation, both alleles would have been cut, and then you will get a 3 kb fragment. What if it was a sperm? What would you expect? In sperm, this allele is supposed to be, it is generally, it is the gene that we are talking about here is maternally imprinted. And therefore, in the sperm, because it is paternal, it will be unmethylated stage. And so you will get one 3 kb fragment like this. Okay. And similarly, I hope you will be in a stage to build up or answer for egg as well. In egg, if it were egg and BGL2 plus not 1, we would get one 5 kb fragment. Okay, so this is complicated, and that is why I'm spending almost the whole hour just to discuss one problem. And it is complicated because you have to think about multiple things. I'll just try to tell you right here. So what we have to remember multiple things. It is about imprinting, right? We have to know which whether it is maternally imprinting or paternal. We should know that. We should know about the map, restriction map. When you say restriction map, it means what are the sizes, where are they cut, and the series of those things, what fragments form, and so on. We should know about the stage from which the sample has been collected, whether it is from somatic cells or primordial germ cells or gametes. And you should also know about the probe. Should know where does the probe bind, the homology or complementarity, based on which we will be able to make the result. So in a typical question, either a gel diagram could be given to you and ask you to interpret it, or a words could be given to you. In words, the description of what is there, and you will have to draw the gel diagram. So either way, you would have demonstrated that you have understood this the or you are in a state to interpret the results. Okay. So now we will move on to something else. If you have any questions, we can discuss on it for 5 minutes at least. Any questions on this? If not, you can always go watch the video again, and we can get back to me in the next class and ask questions. Okay. So there is a different there is a different question. There is a different part of it which I also want to discuss. There is another experiment. Yes. So until now we have discussed about imprinting patterns, whether DNA is methylated or not. Okay. What we should also discuss in the part is how is the chromatin organized. As we discussed before, we can have euchromatin and we can have it as heterochromatin. Heterochromatin is much more compact like this. Euchromatin is accessible by the proteins of transcriptional machinery, so we can expect that there is gene expression from them. Heterochromatin are largely silent. And if you want to analyze whether a particular part of a chromosome is a particular part of the chromosome is in heterochromatin stage or in euchromatin stage, we can use some techniques, and that is what we will learn now. It's a simple one, basically. We know that DNA methylation can induce local histone modification, and that local modifications can change chromatin structure, right? We have seen that DNA methylation attracts some proteins which will attract other proteins that can change the histone or modify the histone tails, mostly methylation and deacetylation, because of which it becomes silent. And then further remodeling complexes will make the chromatin much more compact. Well, in this part of the genome, if you see, there are 4 nucleosomes, just as a representation. By the time it completely becomes, now you have about 6 of them, and the expression is inhibited. So the technique goes by this. And before we'll go, we will also talk about a few things just as a reminder. We have core histones and we have linker histones. H2A, H2B, H3, and H4 are the core histones. H1 is the linker histone. So when we have histones like this, these are the histones in between. And this DNA that is present here is called the linker DNA. That is where H1 binds and makes it a little bit compact. So if I add any enzyme, something like a nuclease, that degrades DNA or breaks DNA. And it can act on the DNA that is freely accessible, something like this linker DNA. It cannot, when the DNA is wound around the histones like this in the nucleosome, then this DNA is not easily accessible by some of the nucleases. But the linker DNA is vulnerable to be acted upon by nuclease. Okay, so when you take chromatin along with that of the DNA and the histones, and you add nuclease to it, the linker DNA, this is where the nuclease acts upon, and then what you have is something like free nucleosomes. And from this, you can isolate the DNA and analyze. So we are going to use this. What we want to study is if a chromatin is in euchromatin stage or is it in heterochromatin stage, right? As you can see, in the euchromatin stage, the linker DNA is long, or there are frequently or frequent linker DNAs compared to that of the heterochromatin, where everything is so packed up. In that sense, if you look at if you add nuclease, the nuclease will cleave this DNA here. So that means they are kind of hypersensitive sites to the nuclease. So euchromatin will produce cuts, whereas heterochromatin, the nuclease may not be able to cut the DNA. So compare this with the technique that we have used a little while ago, where we used is whether restriction endonucleases can cleave DNA or not is dependent on the methylation status of the target site. Here, it is about accessibility to the enzyme. So this is what we are trying to understand now. So what is typically done is, or we will just try to understand what is meant by hypersensitive regions. Hypersensitive in the sense whether to DNAs. Okay. So here is a chromatin or chromosome along with the histones, and then we first treat it with DNAs. We treat it with nuclei. Sorry, DNAs. Treat the nuclei with DNAs, and we have two sites. One is inactive, and the other one is active. Where to mean that gene expression is happening, which is to say, assume that this is euchromatin. That means accessible to transcriptional machinery, and therefore there is action. And then inactive is, we will consider it as heterochromatin. So when we add DNAs, when we treat the nucleus with DNAs, nucleus cannot do anything in this site because it's completely occupied by the nucleosomes. Whereas in the other one, in the euchromatin one, there is a gap here. And that is the linker DNA that is long enough to be accessible by the DNAs, and therefore a cut would be made. And the rest of the DNA is protected because there are nucleosomes. And what we do then is to remove all the protein, meaning that we would have purified the DNA from the rest of the proteins. And now we would do electrophoresis or go through the southern blot technique that we have discussed. That means we would have run through the electrophoresis and we would have used a probe and see what fragment would it form. Because there is no cut here, then it will give the cut restriction endonuclease we have used now to cut the genomic DNA into pieces in this stage. So it will give a 13 kb fragment, and that is what will show up in the blot. Whereas in the euchromatin one, because the DNA is cut here, and later we are cutting the restriction endonuclease, we have a 7 kb fragment and a 6 kb fragment because this is cut here. And when we run the electrophoresis, we are going to see a 6 kb fragment, and 7 kb fragment is not visible. Can anybody answer me why? It's a simple question though. Anybody?
So because of the probe, which it is only in
Yes,
6 kV.
that's right. The probe is homologous or complementary to the region here. If we had a probe, that is something like this, then probably both the fragments would have been recognized, but are visible during the after the development of the blot. But that's not the case here. So that means what it means here with this setup at this point is this region has a hypersensitive site for nuclease activity. That means when you run the gel together and you say like this is A and this one is B, we can say that in this locus, in the locus of the question that we are seeing here, B is much more euchromatin compared to that of A. That is what you can say. Okay, so you can figure out some things. The protocol is something like this. You take the nuclei, you isolate nuclei. You're not isolating genomic DNA directly. Like in the other ones, we would have isolated DNA directly. And then we would have incubated with DNAs. So we'll put DNase protein, which could then go inside, diffuse in, and the DNA is, or chromatin is present along with nucleosomes, wherever possible. The nuclease would have cleaved it, and it would make small breaks like that. And then we are going to purify the DNA. And then we are going to treat the DNA with the restriction endonuclease, or you can say it as restriction digestion, using BGL2 based on whichever, if you're looking at locus, and you will use the restriction endonucleases that are present in that has the restriction sites at that place. And then we would have done the southern blotting along with the probe for hybridization. Okay, that is how we will be able to figure out if there is any reaction site, hypersensitive sites or not. Okay, I think if you have any questions, I would like to take. Okay, I will just answer this one question before we go ahead. So this is how the gels, to look at these gels is actually not so easy, in the sense that one has to require a lot of experience in trying to analyze these results. Say, because we haven't done it, we should go by what the authors say. And I just want to use this gel diagram so that you will be able to understand it as such. In the other ones, usually in the other gels, usually we see a band like this. And this band here is little fuzzy and spread out like this. One of the reasons could be that is you have a fragment like this. Restriction endonucleases are very clear, and they will cleave only in one of their particular site, and they cannot cleave anything else. Okay, but we have used nuclease in this in the gel in this particular technique. And in that one, the nucleases are non-specific. They might cut deep inside. Some of them may not be able to cut deep inside based on the three-dimensional organization of the chromatin at that location. So it does not form a typical band, something like this. Rather, it might form something like this. Okay. So these are some of their results, and this is how they would look like. And we don't have to bother about their results. All we need to bother about is our understanding of the paper. Of the technique, because that's the skill that we are looking, that we need to understand at this time. If you have any questions regarding any of the techniques, please let me know. Otherwise, the class is over. And for sure, I'm going to give one question, the one that you're seeing right now. about analyzing the imprinting, and it is going to be as much as it could be somewhere between 5 to 10 marks if it is an exam for 50 marks in the coming CAA. So please practice this. And the best way is to discuss among yourselves. You can make up questions on this and discuss with each other. Okay. When you make the question, I think that is a good one. If you made a valid question, that means you should have known the answer for it, or you have tried to know the answer. To get a particular or specific answer, you have phrased the question. Then that is even better. So the easiest way to learn is for these kinds of interpretation and logical thinking or deduction, it is better to frame questions or reach a level where you can frame questions which will give one specific answer, and that will be a nice skill. Okay. If you have any other questions, I would like to, any questions at all? No questions?
No, sir. Should be clear if we watch the video. Even if
Yes?
there's no issues, sir.
Okay,
Should
okay.
be clear when we watch the videos again, though.
Yes. So I'm going to send the CAA marks now. Okay, I have corrected. Have a look at them, and if you have any questions, you can get back, and then I will discuss. So I'm not going to comment on the performance. It's only we only have way upward to improve. So we will all work together and improve. Okay. Let's see. Thank you very much.
Thank
Thank
you,
you.
sir.
Yes. Bye bye.
Thank you, sir.
Yes.
Thank you,
Bye.
sir.
Thank you, sir.